Now, let's move on to our next topic, which is osmosis. And specification number 5 asks you to understand that osmosis is defined as the net movement of water molecules from a region of higher water potential to a region of lower water potential through a partially permeable membrane. Water diffusion, or osmosis, is unique because water can pass through most membranes, while many solutes, such as sugar, cannot. Water tends to move toward areas with lower water concentration and higher solute concentration. In general, the cell membrane permits the movement of water. To better understand osmosis, let's examine the given diagram. On the left side, we have pure water, which contains a higher concentration of water molecules and, therefore, a higher water potential. On the right side, there is a water solution with more solute particles, resulting in a lower water potential. A semi-permeable membrane separates these two compartments, allowing only water molecules to pass through while blocking larger solute molecules. Consequently, water moves from the left side, which has a higher water potential, to the right side, which has lower water potential, in an effort to equalize solute concentrations. The fundamental definition of osmosis is that it is the movement of water molecules from an area of higher water potential to an area of lower water potential across a partially permeable membrane. For example, in this diagram, osmosis is demonstrated using a container divided by a semi-permeable membrane. This membrane permits water molecules to pass through while preventing larger solute molecules from crossing. On the left side of the container, there are fewer solute particles and a higher liquid level, representing a region of higher water potential. In contrast, the right side contains more solute particles and the same liquid level, indicating lower water potential. Osmosis then occurs as water molecules move from the area of higher water potential, lower solute concentration, to the area of lower water potential, higher solute concentration. This visual representation effectively illustrates how osmosis functions, a crucial process in biological systems such as water absorption in plant roots and fluid regulation in animal cells. Osmosis is the process where water molecules diffuse from a dilute solution to a concentrated solution through a partially permeable membrane. This movement occurs due to a concentration gradient, which forms because of the difference in the number of water molecules present in the two solutions. In living cells, the cell membrane acts as a partially permeable membrane, allowing water to move in or out depending on the surrounding solution. If a cell is placed in a solution with a lower solute concentration, higher water potential, water will enter the cell by osmosis, causing it to swell, and in extreme cases, it may burst. Conversely, if the cell is in a solution with a higher solute concentration, lower water potential, water will leave the cell, causing it to shrink, which can damage the cell. In animals, maintaining water balance is essential to prevent cell damage. This balance is monitored by the brain and regulated by the kidneys, ensuring that the right amount of water is retained or excreted. And, before we move forward, I want to briefly introduce reverse osmosis, which, while not our primary focus, will help enhance our understanding of osmosis. The provided diagram visually compares these two processes using a U-shaped tube divided into two sections, each featuring a semi-permeable membrane in the middle. On the left side, representing osmosis, the tube contains a diluted solution with fewer solute particles and a higher water level on one side, while the opposite side has a concentrated solution with more solute particles and a lower water level. Water naturally moves from the diluted solution to the concentrated solution, following the osmotic gradient, as indicated by the arrow. This movement occurs due to osmotic pressure, which drives water toward the area of lower water potential. On the right side, illustrating reverse osmosis, we see the opposite process. Here, the left side of the tube contains fresh water with a lower water level, while the right side holds seawater, which has a higher water level and a higher concentration of solutes. Instead of following the natural osmotic flow, external pressure is applied to the seawater side, forcing water molecules against the osmotic gradient, from the concentrated solution to the fresh water side. In essence, this diagram contrasts the natural movement of osmosis with the human-engineered process of reverse osmosis, demonstrating how external pressure is used to purify water by forcing it through a semi-permeable membrane against its concentration gradient. Understanding this distinction helps reinforce the fundamental concept of osmosis and its significance in biological and industrial applications.